Hello everyone and welcome to today's webinar. I'm Eunyoung Lee and I'm delighted to have you with us as we explore life cell imaging and analysis, uncovering cell migration and death mechanism with Celloger. Thank you for taking the time to join us. We have an exciting agenda lined up for you today. We will explore the importance of life cell imaging in research and how it provides critical insights into cellular process. Then we will introduce our advanced automatic live cell imaging system Celloger, highlighting its features, analysis capabilities, and different models available. Finally, we will showcase key applications of Celloger, including wound-healing assays, apoptosis assays, spheroid cytotoxicity, and NK cell killing assays. Have you ever thought about how scientists observe living cells and their activities in real time? That's where live cell imaging comes in. Live cell imaging is a technique that uses specialized microscope and labeling method to observe and study biological process in real time within living cells or organisms. Unlike traditional fixed cell imaging, which only gives you static snapshots, live cell imaging, like you see in these videos, lets scientists watch cellular behaviors like cell division, migration, signaling, and interactions with other cells or molecules as they happen. This approach has become essential in a lot of fields like drug discovery, developmental biology, neuroscience, and cancer research. This graph shows the number of publications each year on Google Scholar and PubMed when you search for the keyword live cell imaging. Live cell imaging technology started being introduced in the 2000s, and since then, the number of scholarly articles on the topic has steadily increased every year. This trend highlights the increasing use and importance of live cell imaging in driving forward academic research. Let's take drug toxicity evaluation as an example to highlight the advantage of using a live cell imaging system. Typically, MTT or MTS assays are the standard methods for assessing drug toxicity. These assays give you quick and rough results, but they only provide information on cell viability at single time point. Although you can set multiple time points, this approach requires preparing separate samples for each one, which greatly increases the number of samples needed and complicates the evaluation of viability across different concentration and time points. In contrast, using live cell imaging for testing drug toxicity lets you capture images as multiple time points for each concentration within the same experiment. This not only allows you to visually observe cellular changes due to the drug, but also lets you analyze cell viability over time across different concentrations based on the image you've captured. As a result, this method gives you a more comprehensive and dynamic understanding of the drug's effects on cells. When live cell imaging technology was first introduced, it wasn't as advanced as it is today, so researchers had to rely on manual methods. This means regularly taking cells out of the incubator, observing them under a microscope, recording the results, and then returning the cell back to the incubator. This repetitive process requires a lot of labor and often leads to significant variability in results between different researchers doing the same experiment. Plus, constantly removing and returning the cells disrupted the cell culture environments and increased the risk of contamination. To solve these issues, automated live cell imaging equipment has been developed more and more in recent years. Our system Celloger features time-lapse imaging capabilities that automatically capture images, ensuring consistent results without variability between users. It can also be placed directly inside the incubator, allowing experiments to be conducted under stable conditions, minimizing the risk of cell contamination. The automatic live cell imaging system Celloger comes in four different models: Celloger Nano, Mini Plus, Pro, Stack, as you can see in the image on the right. With its compact design and efficient live cell imaging capabilities, it has received various certifications and awards as listed below. One of the great features of the Cellular series is that you can place it inside an incubator while still being able to monitor cells in real time from an external PC or laptop. This setup lets you run experiments while ensuring that cells remain in a stable culture environment, free from any external interference, leading to more accurate and reliable results. Plus, you can easily switch out the vessel holder to use different type of vessels like dishes, flask, well plate, and slides as shown below. This flexibility makes the Cellular series versatile and perfect for a wide range of experimental setups. Except for the Nano model, all Cellular products have a moving camera positioned below a fixed vessel stage. In most live cell imaging systems, the camera stays fixed while the plate moves during imaging. As shown in this video, the movement of the plate can cause the medium to shake. If you're imaging multiple points repeatedly, this can also cause the cells to shake, leading to loss of focus and potentially affect their growth. But with cellular products, the moving camera and fixed plate allow for stable imaging of multiple points without losing focus, as shown in the video below. This is a significant advantage, especially for imaging suspension cells, which are prone to movement in the medium. The Cellular software is highly user-friendly and intuitive, making it easy for users to learn and operate. When you're scanning image, the autofocusing function makes it super easy to get the perfect focus. As shown in the video below, you just need to press the autofocus button and it automatically adjusts the best focus. Plus, if you're capturing images at multiple time points, you can set the software to autofocus at each cycle, ensuring clear image over a long period without any focus issues. For those working with 3D structures like organoids or spheroids, you can take multiple images along the z-axis to get the best possible image. If you want to observe changes over time, you can set a schedule to capture images in a time-lapse sequence. Since the device can be placed inside an incubator, you can capture images at your desired time points over extended period without any issues. As shown in the video on the right, when you need to watch rapidly changing cells or organisms, you can use the preview record functions to get real-time video data instead of just capturing still images. After capturing cell images, you can analyze them in a variety of ways using the cellular analysis software. The software comes with annotation tools that make it easy to mark image details like cell size, count, intensity, and scale bars. One of the most commonly used feature is measuring cell confluency, which shows the percentage of the surface area covered by cells, and fluorescence intensity, which measures the brightness of fluorescence signals. By uploading images taken at different time points and clicking the analysis button, the software automatically tracks changes in cell confluency and fluorescence intensity. You can easily extract this quantified value as raw data or represent them as graphs. This makes it simple to analyze cell growth, gene expression levels, and toxicity using fluorescence markers. When you need to view a large area at once, the stitching function allows you to combine multiple images into a single comprehensive image as shown below. And for image capture with the stacking in the scan software, you can even create a video as demonstrated. And the G projection function also lets you combine fluorescence images into a single optimal 2D image. You can also specify a ROI to measure fluorescence intensity within that area or automatically count the fluorescently expressing cells. These features, which are included as part of the standard package, make Celloger software a powerful and versatile tool for a wide range of cell imaging applications. As I mentioned earlier, the Celloger series includes 4 models, and the Celloger Mini Plus is a key device in the lineup. It features a fixed fluorescence filter and magnification, making it reliable basic live cell imaging experiments using the scan and analysis software we discussed earlier. This setup ensures stable and accurate results across a variety of applications. Next, the Cellular Pro is an advanced model in the lineup, offering greater flexibility and enhanced features. It lets users change the lenses themselves, so you can adjust the magnification to fit whatever you're observing. Additionally, with 2 fluorescence channels, you can observe multiple targets at the same time, which opens up a wider range of experiments. And the Cellular Nano is an ultra-compact, lightweight digital microscope, roughly half the size of an A4 sheet of paper, making it highly portable and versatile for various settings including incubators and clean benches. It images a single position with precision and offers time-lapse scanning for in-depth observation. With X, Y, and Z axis knobs, it allows for quick and accurate focusing, making it an ideal tool for precise and flexible cell imaging. Lastly, the cellular stack serves a slightly different purpose than the other 3 models. It is mainly used for large-scale cell culture processes using multi-layer vessels. With its time-lapse and multi-position imaging and analysis features, it helps you determine the best time to harvest cells and establish standardized protocols by setting the ideal culture condition during the process. Now let's dive into the various applications of the cellular system. As you can see here, we've listed some of the key applications that have been successfully carried out using the cellular series. With Celloger, you can monitor cell movement and morphology, cytotoxicity, and cell function in real time across a wide range of experiments. Today, we will take a closer look at some of the most important ones, starting with the wound healing assay. The wound healing assay is a fundamental technique used to study cell motility and cell-to-cell interaction, especially in fields like cell biology, cancer research, and tissue regeneration. This method allows researchers to visually track how cells migrate to close the wound, offering valuable insights into cell migration ability, proliferation rate, and the impact of various treatment or environmental conditions on cell movement. Real-time imaging greatly enhances this assay by providing accurate measurements of wound area reduction, cell migration distance, and wound closure speed. Traditionally, this process involves manually observing and capturing images of the wound area at regular intervals under a microscope. However, with the automated lifestyle imaging capability of Celloger system, continuous manual checks are no longer necessary. The timeless function automatically captures images at set intervals, and the analysis software make it easy to solely examine the collected data. Here, the video on the right demonstrates the wound healing assay conducted using a Celloger Mini Plus with 4 different cell lines. Each cell line was seated in the same well plate and imaging was carried out simultaneously. The images were captured every 30 minutes over a period of 3 days. With the cellular analysis software, analyzing the wound area is fairly simple. First, just drag the image you took with the cellular scan software, and then from the top menu, choose wound area under the Analysis tab, and you will see the wound area highlighted in yellow, like you see here. After that, click the Analysis button on the right, and the software will measure the wound area for each image and show the result in the wound area section on the right. If you want to see how the wound area changes over time in a graph, just click the chart button and the graph will pop up. The dots in the well position window show the points that were imaged, and clicking on any dot will display the graph on the left. Finally, if you need a raw data, just click the Export button at the bottom to download it as an Excel file. This easy process makes it quick and simple to analyze wound healing experiments, which are widely used in various fields. This data, published in 2022, shows changes in gastric cancer cell migration observed through a wound healing assay using Celloger. As you can see in the experimental scheme, MKN1 gastric cancer cells were fully grown in a 24 well plate and a scratch was made in the center. And cells were then treated with cancer-associated fibroblast, which is calf serum, where E2 signaling was inhibited, and the rescue group, CD147, was additionally applied. And the cells were imaged for 48 hours using the Celloger Mini Plus. As shown in the image, the group treated with E2 signaling inhibited calf serum exhibited less cell migration compared to the control group. However, when CD147 was added, cell migration was restored to level similar to that of the control group. The captured images were easily analyzed using cellular software, making it simple to measure cell migration as demonstrated in the graph. Next, let's explore how to use live cell imaging to observe one of the key cell death mechanisms, apoptosis. Apoptosis, which is programmed cell death, is a carefully controlled process that includes events like membrane blackening, cell shrinkage, and nuclear fragmentation. Since Apoptosis is closely tied to cancer development. It's a big focus in the development of new anti-cancer drugs, and it's often used to assess the toxicity of potential drug candidates. Typically, when running an apoptosis assay, researchers target the annexin 5 marker and use endpoint methods like flow cytometry. But these methods require several steps like fixing and washing the cells and only give you a snapshot of apoptotic cells at a specific moment in time. With live cell imaging, you can easily track the percentage of apoptotic cells over time with just a few simple steps. Plus, you can actually see the cell shrinking and changing shape as they go through apoptosis. When conducting an apoptosis assay using live cell imaging, researchers commonly use a caspase-37 detection dye. As shown in the image on the left, caspase-37 play a crucial role in the apoptosis pathway, and this reagent is a dye that becomes activated by caspase-37. This reagent includes a green dye attached to the DVD sequence that caspase-37 recognizes. When you treat cells with an apoptosis-inducing drug along with this reagent, caspase-37 get activated and cleave the DVD sequence. The cleaved green dye then enter the nucleus and bind to DNA and emit green fluorescence, signaling that the cells are undergoing apoptosis. Here, HeLa cells were treated with the apoptosis-inducing drug staurosporine and simultaneously stained with caspase-37 green dye. Using the Celloger Nano, we captured images every 30 minutes over a 15-hour period. As you can see in the image and the video below, the number of cells showing green fluorescence increases over time, indicating that more cells are undergoing apoptosis. Additionally, in the bright field image, you can clearly observe the cells shrinking and changing shape as time progresses. With the cellular analysis software, you can easily measure the level of apoptosis over time using fluorescence analysis. Just upload the time-lapse images captured from the cellular scan software into the analysis software. Then click the green light button at the top right, and then click the analysis button. The software will automatically measure the green fluorescence confluency and intensity for each image, showing the result on the right. If you click the chart button, you can generate graphs that show how the fluorescence confluency and intensity change over time. Finally, you can click the export button to download all the data as raw Excel files. In this experiment, we measured apoptosis over time by tracking green fluorescence coverage as shown in the graph on the right. You can see that the fluorescence starts to increase rapidly around 3 hours after drug treatment. What I just talked about was checking cytotoxicity in 2D cell system. But these days, the trend is to use 3D spheroid or organoid for drug toxicity testing. You can use Celloger to run experiments on these 3D cell model as well. Drug development often starts with 2D cell cultures in vitro testing before moving on to in vivo animal models. But because of differences in the cellular microenvironment, the results from in vitro tests often don't translate well to in vivo conditions. With the growing concern for animal welfare, there's also a push to reduce animal testing and find alternative methods. That's why 3D cell cultures, especially spheroids, are becoming more popular in drug screening and toxicity testing. These 3D cultures help bridge the gap between 2D and in vivo models, better mimic tumor environment, retain tumor heterogeneity, and provide a solid alternative to animal testing. The cellular is really useful in this process, making it easy for researchers to verify the formation of spheroids and conduct precise cytotoxicity tests. For spheroid formation, there are various methods available, but we choose to seed the cells in 96-well U-bottom plate. In these U-bottom wells, the cells come into contact with each other and gradually aggregate into round shapes, forming spheroids. This process can be clearly seen in the video below. The video shows a comparison of spheroid formation using two different cell types, MDMB231 and HeLa, with varying cell seeding numbers. These images were captured every 15 minutes using the Celloger Mini Plus. In this experiment, we used cancer spheroids to check the real-time toxicity of an anti-cancer drug. HEC-293 cells tagged with GFP were seated in a 96-well U-bottom plate to form spheroids. Then we added the anti-cancer agent cyttosporin at concentration of 0.1 and 1 micromole, along with ethidium homodimer 1 dye, which stained dead cells. We captured time-lapse imaging using Celloger Pro. As you can see in the image on the top right, the red fluorescence intensity increases over time after drug treatment, with higher concentrations showing more intense red fluorescence. The timeless video on the bottom left really emphasizes the differences between the control group and the drug-treated group, giving a clearer view of how the drug affects the spheroids. By measuring the red fluorescence intensity with Celloger's fluorescence analysis, we were able to quantify the spheroid viability and display it as a graph as shown on the right. Additionally, the spheroid size decreases as the drug concentration increases. When using the cellular analysis software, there are two main methods for analyzing spheroids. The first method involves using the ruler tools in the basic analysis section to measure and analyze the diameter of the spheroids. The second method focused on analyzing spheroid size by measuring its area. The software automatically detects the spheroids and calculated area for you, which is quite similar to the wound area measurement process. To do this, simply upload the timeless images captured with the Cellular Scan software. Then from the top menu, select Spheroid cell under the Analysis tab, and the spheroid will be highlighted in yellow as shown here. After that, click the Analysis button on the right, and the software will measure the spheroid area for each image, displaying the result in the area section on the right. You can also export the raw data, which allows you to quantify the spheroid size based on the drug concentration. Lastly, let's take a look at the NK cell killing assay. The NK killing assay is a way to measure how well NK cell can kill target cells like cancer cells. When NK cells interact with cancer cells, they activate receptors on their surface that trigger the release of cytokines like interferon gamma or TNF-alpha, as well as cytotoxic granules like granzyme B or perforin. These substances work together to kill the cancer cells in a non-specific manner. With the rise of cell therapies, especially CAR-NK cell therapies that target cancer, NK activity assays have become an important tool for evaluating effectiveness of these treatments. There are several ways to measure NK cell activity, but here I will explain how to use live cell imaging to check NK cell activity across different target cell types, whether they are suspension cells, adherent cells, or spheroids using cell death markers. First, we tested cytotoxicity using K562 cells, which are the most commonly used target cells in NK killing assays. K562, which is suspension cells, were stained with carcine AM, which emits green fluorescence. Then we co-cultured these target cells with NK cells at a ratio of 5 to 1, 10 to 1, and 20 to 1, and added ethidium homodimer 1, a red fluorescent dye that stained dead cells. We took images every hour for 24 hours using Celloger Pro. Over time, you can see the red fluorescence getting brighter and green fluorescence getting dimmer. And as you can see in the video below, as more cells die, the clump where NK cell and dead target cells gather together became more frequent and larger. For adhesion target cells, we conducted the experiment using U2OS cells. The process was pretty similar to what I described earlier. We stained the U2OS cells with cell tracker CMFDA dye, which emit green fluorescence, and then co-cultured them with NK cells at ratio of 5 to 1, 10 to 1, and 20 to 1. After that, we added ethidium homodimer 1 dye to stain the dead cells and started time-lapse imaging right away. Just like with the suspension target cells, we saw that over time the red fluorescence got brighter and the green fluorescence faded. Here we use Celloger's fluorescence analysis to compare the result more quantitatively. We measure the intensity of each fluorescence. To determine how much cell death occurred, we divided the red fluorescence intensity, indicating dead cells, by the green fluorescence intensity, representing the total number of cells in each image. To focus on the cell death specially caused by the NK cells, we subtract the amount of spontaneous cell death that occurred without NK cells from the result. By applying the formula shown on the right, we were able to track NK cell-induced target cell death over time. When you look at the result from the experiment with suspension target cells, you can see that the amount of target cell death increases as the NK cells ratio goes up. On the other hand, in the experiment with adherent cells, the target cell's death rate caused by NK cell is relatively lower. This might be because suspension cells have a larger surface area in contact with NK cells compared to adherent cells, which likely makes them more affected by NK cell activity. You can run experiments not only with suspension and adherent target cells, but also with spheroid models as NK cell targets. We formed spheroids using GFP expressing U2S cells, then co-cultured them with NK cell at different concentrations. Just like before, we stained the dead cells with ethidium homodimer 1 and used the Celloger Pro to capture image. As you can see the image and video below, The target cells form a ring along the boundary where the NK cell contact the spheroid. The higher NK cell concentration, the thicker this red ring gets. As a result, we can see the green fluorescent spheroids shrinking as the target cell die off along the edges. In 3:1 ratio, NK cells clusters along certain part of the boundary, and cell death was especially noticeable in those areas. In addition to the application we've covered so far, Celloger allows you to dynamically observe various cell types across a wide range of experiments as shown below. If there's something I didn't mention today, please feel free to contact us. We'd be more than happy to discuss the different application with you. To sum up, Cellular allows scientists to capture and measure different cell processes as they happen in real time. Whether studying wound healing, cell death, tumor growth, or immune cell activity, Cellular provides clear and detailed information about how cells function and interact. By making complex experiments easier and giving researchers valuable data, this innovative tool not only saves time and effort, but also opens doors to new discovery in cell biology. With Celloger, scientists are better equipped to unlock the secrets of cellular life, potentially leading to breakthroughs in medicine and biotechnology. Thanks so much for joining today's webinar. We'd greatly appreciate if you could take a moment to fill out a short survey. You can do this by scanning the QR code on your screen or using the link we've just posted in Zoom chat. Additionally, we are offering you the chance to sign up for a free trial of our scan and analysis software or request a free demo of the equipment. We would love for you to take advantage of these opportunities. Thanks again for being with us today. If you have any question about the webinar, please feel free to drop a message in the Zoom chat. We are here to help.